Greetings, everyone. This is Dr. Ross, and we're very excited to have him here today, and it's very gracious of him to give his time. Well, Dr. Ross, I think the first question we would like to start off with is, given that you've seen these results in one session, I believe this question was in regards to your Mind Over Chatter article. What are your thoughts about whether or not this method may provide an experimental model for testing whether some forms of neurofeedback are better than others?
Okay, long question. But so yeah, thank you very much for inviting me. I'm just very happy to talk about neurofeedback with you, and also that it's for the ISNR that I consider the spiritual home of neurofeedback. Since it came from the US, and it's a society that also has made sure to retain the flame for long. And I hope that there will be more scientific studies coming out soon. So yeah, to your question, basically, I think it's a good model to assess neurofeedback results directly, because it's a short-term effect, and so there's less variables to sort of consider when one is looking for the effects, because it's immediately after a session that hopefully the differences are due to the actual entrainment of the EEG, and the correlations are quite robust with some measures that one can be confident it's really due to the training if one finds them. On the other side, it's not such a good model for obviously long-term effects, and it doesn't supplant those models, because obviously all neurofeedback requires many training sessions, because the training is gradual, which is the advantage of neurofeedback because of the safety. The disadvantage is it takes longer. So I would say it's a good model, but I think it's highly dependent on how well the person trains, and with many neurofeedback protocols it requires some time for people to learn to control their rhythms. So I think until it's demonstrated that people can harness a certain protocol, then long-term training is, I would say, necessary. But if once somebody can demonstrate that they can, let's say, control the rhythm quite well to be able to increase the signal-to-noise ratio within one session, like I've been using with this one protocol, because it's easy to learn, but with other protocols that might not be the case. So basically, what I mean is that you would need to have well-defined, training efficacy, and then see, okay, is there an effect? But of course, there's some other effects that may be also downstream to that. So even sleep, there may be some changes occurring. So I would suggest the same as with other types of neuromodulation research, that there's both, immediate effects that are being examined immediately after the session, and also effects maybe after sleep and after many training sessions. That's it.
All right. Thank you very much. That was a very thorough response, and it sounds like there are a lot of opportunities in there for more exploration, definitely, and more research. So.
Sure. I think a sort of a two-pronged approach, where we looked at the short-term effects and long-term effects, that will have a comprehensive vision of what's going on. Obviously, the short-term effect is that it takes much briefer period amount of time to implement, because it's only one training session, and that's one of the reasons why I've gone that way. Because in science, I remember my first study took me 4 years to do, and it was many neurofeedback sessions per group. And so now in the scientific climate, it's publish, and get as many papers out. So that's also a reason why it's nice to do immediate effects. But the other reason is that I've been finding that in some protocols it's not so easy to learn immediately. So I think the one workaround to that is that you could you could pre-train the person, or select some candidates who can do some protocol, and then do a short-term training effect on that subset, or after you've trained them, then give them a pre and post session. So that way you save time and identify the people who can do it, if you wanted to look at neurofunctional correlates over some training method.
That's certainly interesting. Yeah. All right. Well, our next question is, do you know if FZ alpha down training has been used in clinical conditions such as ADHD or attentional problems secondary to trauma? And as a second part, do clinical subjects respond differently than normals?
Okay, so with regards to that, actually, the training that we did, the mind over chatter study, it was PZ alpha
Okay.
down. But yes, of course, the main site of sort of modulation, what we found was the cingulate area, which is in the frontal area, in the frontal lobe, and on the MRI, it was sort of, you could say that maybe there's some kind of frontal effects, and also some of the alpha changes were in the frontal lobe. So it seems like there's some kind of top-down modulation of this parallel rhythm that are maybe frontally driven. So that is a rationale for maybe doing even frontal training directly with alpha. But that hasn't been validated. So the only experience I've had with the PZ alpha down training that's clinical is with Professor Lanius in London, Ontario. Uh, along with doing the healthy subjects, we also ran a study that is with PTSDs, post-traumatic stress disorder. It's not a controlled study, but we looked at the effects of this same protocol on this clinical population, so that's going to be published soon. But what I can say is that there seems to be some effects there that are contrary to what one would expect in terms of what's happening to the alpha. So one of the effects that seems to occur is there's an alpha rebound, which actually increases alpha after the training. That can be due to the fact that their alpha is quite low to start with, and then there's a homeostatic sort of self-correction mechanism in the brain that increases alpha after training, maybe even a fatigue effect. But that seems to be related actually with the calming effect on subjects. So it may be that it has a kind of a secondary effect of calmness, and that's associated with an alpha increase. And also, as you know, there's quite a lot of literature showing that alpha up, so increasing alpha protocols are quite beneficial for PTSD as well as anxiety problems. So that could be one kind of mechanism that occurs. Yeah, and I've personally also been involved in some sessions with Ruth Lanius, with a subject that has that received quite a lot of alpha down sessions, 2 or 3 times a week. It was just one person that we sort of wanted to see, okay, what's the general effect? And she seemed to respond quite well. She later said she had much less anxiety. Unfortunately, we didn't have much pre to post QEG or anything like that. But overall, she improved quite well. So, but this is anecdotal. So, but there might be some beneficial aspect of this protocol for clinical populations. Obviously, for attention, because it improves. It seems to reduce mind wandering. So, my most sort of interesting application, I think, to follow up on would be to see, okay, would this be a new protocol or an additional protocol for ADHD, but also maybe even some other mood disorders. But so far, that hasn't been tested much yet.
That's interesting. That leads us into our next question, which is there any evidence that the changes in connectivity and mind wandering are seen with any of the protocols that have been popular with ADHD?
I have to say, I'm not really acquainted with, I mean, I am acquainted with the ADHD literature, but I'm not acquainted with many studies doing connectivity analysis. So, to my knowledge, to the best of my knowledge, I don't think there is any of that sort. I know Robert Cohen has done some protocols that show connectivity changes in coherence in the EEG level. But as far as the MRI, I don't think there is anything apart from Mario's work that is simply activation changes, no connectivity. So.
Okay. One of the next questions is what happens after a series of training sessions? Has that have you done much research into that area with this protocol or any others?
Well, actually, yeah. So that's where what I previously mentioned. This one patient that we had, one PTSD patient that had a series of trainings, and she felt like she had much better mood control. She was less anxious coming into lifts and taking the train. I remember we talked about it with her. So she seemed to have quite a revolutionary change, actually. Apart from that one subject, Ruth Kanius is continuing to do some research with long-term training, but I don't know at what point they will publish their results on a group level for long-term training. So at the moment we don't know what the real effect is long-term.
Okay. That takes us past our next question then, which had to do with some of the long-term duration training. So the question after that would be, have you thought about collaboration with Mario Beauregard's group at the University of Montreal with his findings of volumetric changes in gray and white matter following a number of sessions of attentional neurofeedback in normals? Some think the case for the utility of neurofeedback relies much more on demonstrations such as these than large randomized control trials of efficacy. What are your thoughts on these topics?
Sure. I mean, I'm always open to collaborating with groups that are interested in looking at neurofeedback, and I know very well Mario's work, and I think it's definitely a piece of the puzzle that, as far as I can hear now from reading the abstracts, that has been validated that there are changes in white matter. So that's very good news, and it's something that is to be expected if there are changes also on the behavioral level. So it complements the functional changes that we see, and of course it needs to be shown that this can generalize to the hardware of the brain, because the brain is kind of like neither software nor hardware, both influences the other. So since when the software changes, the protocol would be considered the software. So as you train a certain type of activity, then also the structure changes, the hardware, and vice versa. The hardware affects the software. So that's both of those elements are important to show that the plasticity occurs. So I welcome that kind of work. The caveat is that it does take a large amount of time, but then the fact that you can prove that these changes occur is a great evidence that neurofeedback has an impact on the brain. And it's been shown in literature that many other types of brain training, whether it's skill learning or mathematics or motor skill learning or anything, that there's very quick changes in the brain structurally after some training sessions. So almost half a dozen training sessions already can implement, can induce changes in the brain. So I think we would have to investigate the limits of that with neurofeedback. How many sessions do we need to use to induce changes, to detect changes? It could be as little as half a dozen maybe. But at the moment, I don't know now with Mario's work how many sessions they have in total. But that would be definitely something that would be an additional picture on where do the effects occur in the brain during occasion for particular protocols.
All right. What are your thoughts about the second part of the question that have to do with your research and Mario Beauregard's? Some people feeling that they're much better of a demonstration for neurofeedback going into the future instead of randomized control trials for efficacy.
Okay, I see. I think they're complementary, and it's obviously good to see a mechanistic counts of what's happening to the neurophysiology of the brain. But even the newest randomized controlled trials, for example, by Heinrich, have been very good because they also looked at neurophysiological measures at the same time. So, the ideal study would be something that incorporates everything. short-term, long-term changes, neurophysiological as well as behavioral, and it's a clinical population. Obviously, that requires a lot of resources, but I think just neurofunctional studies based on healthy populations are not enough to show that a protocol has efficacy on a clinical level. And I think the community should support both types of studies, so that we have a picture of the brain, sort of maybe short term or medium term, and then also, okay, what does this do to the behavior in the clinical setting? So in that way, the two reinforce each other, I think, and that's the same approach is being used for other neuromodulation techniques.
All right, thank you. Can you give us a preview of some of the next research you have in neurofeedback that would be coming through the pipelines, so to say?
Yes, of course. I mean, I'm here now in Geneva, in Switzerland, working on some new applications of neurofeedback. This lab here specializes also in emotions and attention. So we have one project wanting to look at whether neurofeedback can, again in healthy subjects, modulate a sort of affective function, so emotions and so on. But it's quite an open project. So at the moment we're just exploring, what type of protocols can people learn. We've tried to look at the frontal lobe and see maybe modulating theta rhythms and alpha rhythms. And just basically getting a feeling of, okay, what people can learn to do a one-session paradigm, because I've been sort of trying to use that paradigm, but nothing really tremendous that I can speak of at the moment that we found. But also the hope is that we will move on to clinical populations, because there's also a hospital here in Geneva across the road where we work. We should have some access to patients, hopefully. So hopefully we get some access to also major depression, possibly bipolar disorder, and so on. So we'll see. It's difficult to come up with a protocol that is not sort of neurophysiologically based. So the first step is obviously to maybe isolate a group of patients to see, okay, what does the clinical picture look like? Maybe with cognitive EEG or during a particular task, and then I try to motivate a protocol based on that. At the moment, we're motivating protocols based on the EEG literature, but I'm also wary of the fact that patients and healthy subjects are different sort of kettle of fish. So we'll have to see whether one thing generalizes to another, because obviously healthy people are operating within an optimal spectrum. And therefore, okay, you can give them a neurofeedback protocol, but you could technically also, in fact, impair performance if they're optimal to start with. So we're looking at all these effects and then trying to build a model of, okay, what do these frequencies do, and also maybe looking at new neurophysiological EEG parameters, conductivity, and so on. So it's a pretty wide sort of project. So we want to identify new ways of doing neurofeedback. But at the moment, I can't tell you for sure what protocol we're going to decide on at the end to treat patients.
Well, it sounds very interesting. It sounds like there's going to be a lot of exciting news coming out of there.
Well, I hope. You never know. You can be lucky so many times.
Yeah. Well, it sounds like there's a lot of work and dedication going into it. So we're
sure. I think it depends on just you try to do your best to uncover the sort of the underlying truths, and without being too attached to what the result is in terms of your models. And we're finding also that the brain is much more complicated than we thought. So certain models that you hold and say, okay, this goes up, then therefore it will stay up, and it's from my experience recently, the brain is much more of a circular sort of system than just up and down and so on. So we'll see. We're also dealing with some of these homeostatic effects that I mentioned before about rebounds and things like that. That's kind of a new aspect of that I haven't seen in the literature in terms of brain training, or at least neurofeedback.
Yeah. So that maybe kind of leads me into my next question, which is, what advice would you have for earlier career researchers who are interested in pursuing or moving their career towards research into neurofeedback?
Okay, well, I would definitely encourage it. Um, Partly because I think it's a hot new area that has a big potential. That's why I'm in it. But also because the field is wide open. It's in its infancy, so so many opportunities to discover new things. And as a young researcher, you can sort of hope that there is a place where you can find your place. So it's kind of an unexplored territory. The world is your oyster kind of place. So according to those criteria, it should attract young researchers and their curiosity, which is high. And secondly, also because I think it's interesting to see whether we can, treat brain problems with this kind of approach, training approach that's based on neurophysiological parameters. It's philosophically it's a different approach than other ways of treating the brain from the outside, and it's more from the inside, and hopefully that should improve the stability of the effects afterwards. But I would say for young researchers, the advice I would have is the same: is to just have an open mind, not so have a look at what's been done in neurofeedback before. Obviously, build on top of that to make yourself um, well versed in the history of neurofeedback, because there's a lot of literature there that most people say there's not many studies, but there's studies going back to the mid 60s, and a lot of these have been forgotten and have not been cited. So you could do your own literature search and find out, okay, what are the effects? That's one thing, and the second thing is to also take advantage of the knowledge we've accumulated so far that maybe is not necessarily directly related to neurofeedback, but it's been done by the EEG community, or depending on the type of neurofeedback that people will want to research. There's a lot of research that can be situated for the purposes of neurofeedback. So, for example, in EEG, there's been many studies done with alpha or theta and gamma. There's many studies out there. And then the idea is to use that knowledge and translate it somehow into a neurofeedback approach. That's really kind of I would say the key, and also the challenge to do that. Because the scientific community is large, we should take advantage of that, and we're not alone. So we should feed off each other, kind of thing.
All right. Thank you very much. The final question for today then would be, how did you become interested in neurofeedback?
Oh yeah, okay. And so I remember that it was back in 2004, I think. I actually did a physics degree at Imperial College in London, UK. And I remember there was a public demonstration of neurofeedback by Professor Roselia. And I don't know, I was just passing by and I saw it. I was like, oh, this is interesting. It's kind of like meditation, but at the same time, it's like playing a video game. And then somehow I got captivated. And I got in touch with John and asked him if there was a project. And I remember, there were not many leads that were successful for me to keep doing neuroscience after my master's. And luckily, Yes. So yeah, I was saying that luckily, John's lab was considering another research, so the officer was very happy to take it, not knowing exactly where it was going to go, but then as I got into it, I learned more about neurofeedback, and I was very happy to continue doing the research. So it was a stroke of good luck to learn about neurofeedback that way, and then it led into my PhD. So I did a couple of years in neurofeedback research before I started the PhD officially. Um, but they were very, I was very happy to learn about it, and it was difficult at first. I know that once you get into it, it can be very confusing, and there's so many different opinions and models, and then also the difference between the clinical community and the scientific community. So it took a while to orient myself, but I think nothing beats a good experiment of your own, and a lot of the times I actually did experiments on myself. So that way I got some, for example, the mind over channel study was actually directly inspired by some of the meditation work I was doing, the mindfulness at that time. And I just sort of hooked up my EEG and looked at the brainwaves and thought, okay, this is cool, let's see if people can control this. It took a while to get to it, more or less 5 years or something. I more than that, 7 years. But I did another project before them, and then I wanted to also validate neurofeedback and TMS. So I think we're slowly getting traction in terms of getting neurofeedback accepted by the mainstream scientific community, which is the main sort of, I think, the sine qua non, the indispensable part of the equation that needs to be fulfilled for the rest of the scientific community to be, for us to kind of get everybody on board, and then we all work on the same problem. Because at the moment there's too few people interested. There's a lot of clinicians interested, but they're not as publishing as much. And if you want the field, to have innovations and more people doing real experimental science, we need to have other scientists involved that see promise in this method. So I think we're just on the cusp of getting people to see it as to prove it as a concept that it works. So at the beginning of my studies have been more proof of concept. So okay, this is there any change? Yes, there is in the plastic sort of function of the brain and so on. And then after we get people confident that this is as a concept works, then the next stage, which is going to be the much harder one, is to really tailor each protocol for a particular disorder and see how far that can go. But I think, slowly I'm absolutely like that.
That's interesting, and that is very exciting.
Yeah.
Go ahead.
Yeah, there's many more studies, also validation studies to be done. But I think any parts that any type of disorder or any type of approach that people can demonstrate is scientifically viable is welcome. And so therefore, there have been these traditional protocols that are useful and that have shown some promise for ADHD and epilepsy and so on. So that needs to be maybe even replicated, because there have been too few of those studies that the scientific community still hasn't accepted. So there should be first a replication of previous, pioneering results, and secondly there should be innovation, which means build or maybe even explore different approaches to see whether they're more efficacious or more useful for a particular disorder. It could be that one protocol works very well for everything, or it could be that there needs to be tailored protocols for particular disorders. That still remains to be seen.
All right. Well, that concludes all of the scripted questions we had. Are there any topics that you would like to add in or discuss at this point?
I think I pretty much covered most things in this very young field. That there's a lot to talk about, but at the same time there's a lot of work to do in terms of finding out what the effects are. So I'm quite cautious about trying to say something works more than another. And I think even with my own results, I would like to see that they get replicated, so that I'm I'm happy that, okay, some of these things other people can find. It's not just a figment of my imagination, let's say.
Yeah. All right.
I would do that too. Okay, well, anyway, it was a pleasure, Mark.
It was definitely a pleasure from my end, and I thank you very much again for your time, and I hope everybody that views this is excited about this interview as I am.
I hope they enjoyed, and I hope they continue doing some work in neurofeedback.
All right.
So, thank you very much, and have a nice night, and catch you sometime maybe in the future.
Yep. Thank you.